Excuse me. Um, so enzymatic proteins are where I want to uh, start, and then we'll go back and talk a little bit about the carrier protein in specific for details. So these things in the membrane structure that I was showing you was like, for example, an outer membrane of the cell. So this paper here, my outer membrane. Okay. Uh, we're going to talk first about prokaryotic cells, and prokaryotic cells do not have membrane-bound organelles inside of them. So membrane is all going to be kind of laying flat, or just it's extra layers and thicknesses of membrane just right inside the outer membrane is where you're going to have uh, reactions occurring, like for cell metabolism. Cellular respiration is going to go on there. If it's a photosynthesizing uh, cell like cyanobacteria, then you're going to have some of these membranes that have the enzymes needed for photosynthesis. Uh, when I talk about layers of membrane, I'm talking about like this type of thing here. Okay, so if I were a cell and I had to be making, burning sugar, and this was the only area I could do it in, that's my limit of energy I can make. But cells do extra layers, and this is a pattern that you see throughout nature in biology. We'll come back to this type of structure over and over again, and that's folded or stacked, okay, to increase the amount of surface area in a smaller space. So for example, I've got two pieces of paper here, roughly same amount of size, okay, same size with each other. I'm going to take them, I'm going to take this one, I'm going to fold it, accordion, fold it like a fan, okay, and put that inside my cell here to illustrate how, I'll get in there, how I can have folded membrane in a cell to increase surface area, because look what just happened. Now, not only do I have just this inside of the membrane that can have enzymes embedded in it, I can have one side and the other side of this membrane folded up in there to be the site of my cell metabolism, if that's what I'm doing. Because these enzymes have to be present to make these reactions happen. So anywhere where these enzymes are is a site where when these reactions occur, there's going to be a whole various enzymes, okay, needed for those reactions to occur. But we got to have space for that. As long as I have, room in between for some cytoplasm, for the liquid, for things, molecules to move in and around, these can be stacked pretty close to each other. All right, and so by doing this, considering that the inside membrane here really probably doesn't have a lot of room for this to happen, it's going to be the interior membrane. Rather than just having a flat membrane, I've doubled, okay, the amount of space I have for these reactions to occur. And then it's like, do I have room for more? Okay, I could probably pack this down a little bit tighter and still have room for reactions. And then do I have room for more? And yeah, I could put more folds in there. I could put more of these stacked in there, maybe, 2 or 3 more. So that really increases the amount of these types of reactions that can occur inside a cell. So we see that, and when we get into the organelles, you'll see that inside the organelles, the mitochondria and the chloroplast both have this type of folding or stacking structure going on. In our lungs, we have alveoli, get that out there, which are little sacs that kind of stick out from the membrane lining of the lungs that increase the surface area for gas exchange, because the gas has to exchange across the membrane. And in our intestines, we have these little villi that stick out that help absorb water and nutrients out of our intestines. Okay, so, and I've got a slide on that here in a little bit. So fish gills, when we get to fish, we'll talk about gills a little bit and their structure. It's the same, that surface area, increased surface area. So that's a pattern. I'll talk a few times here over the semester about patterns of nature, things that are just kind of, no matter where you're at, this is the pattern you're going to see repeated over and over again for efficiency. And this is one of those efficiency patterns. So That's where you're going to find your enzymatic proteins is in your internal membranes of the cell, either just inside that outer membrane or inside organelles if it's a eukaryotic cell. I've already talked about this slide on Monday as far as cellular fingerprints and talked about these functions here, what can go through and not go through easily. So that's all repeat, or actually, I guess probably this is imprint of what I said. So you can refer back to it and study it. Excuse me. So Landis, I'm gonna sneeze going on. Okay, so diffusion is where you have something in a higher concentration and it moves to an area of lower concentration. And the analogy I like to use on this is if I were in the classroom and I were to close the two doors at the front and to go to one corner and spray an air freshener or perfume or cologne or something that smells, I'll use air freshener as an example though, spray it there, it would distribute eventually throughout the room. So we shut the doors. Let's say the air unit doesn't kick on the whole time we're doing this. We've got it turned off. Molecules bounce into each other and they reflect off of each other. And that natural movement that just happens is going to, without air currents, is going to cause that um, chemical aromatic. That's what you smell when you smell any type of perfume or cologne or air freshener or anything. Those are aromatic chemicals. Those that aromatic is going to get distributed around until eventually every place in that room has the same concentration of that aromatic. So before I did that spray, I took a sample, several samples throughout the room and tested for the presence of that aromatic, it wouldn't be there. If I tested right as I sprayed, that aromatic would only be in the corner and it wouldn't be anywhere else in the room. But over time, you would see the concentration at corner would decrease and you would start seeing it in other samples from the other locations of the room. And eventually, all of the samples that I would take in the room would have the same concentration of that aromatic found in them as it equalizes. It establishes equilibrium. Okay. And it would stay that way. It would just stay that way in that room. Uh, unless something changed, like we opened the door at the end of class. And now previously people in the hallway wouldn't be able to smell it, but we open the door and now those particles are going to move out. We open the door and we move out in the hall, we're going to carry that smell with us. But if we were to open the door and just sit there, it would still eventually drift down to the hall. Okay. Without air currents carrying it. So that's your diffusion. Now, if we take that process, we look at water and we put it across a membrane, then we have osmosis. Okay, osmosis is net movement of water molecules from a region of higher concentration to an area of lower concentration across a semi-permeable membrane. So sometimes you'll see in books, and you may have learned this may have been a simple definition given to you in life sciences or biology class earlier on, that osmosis is diffusion across a membrane. And you can think of it that way. Okay, it is water movement though. So here the example is what I was mentioning just a few minutes ago about the process of water being absorbed from human intestine, taken up by the body. And I'm thinking I have said this some weeks back, but I'm saying it again in case I didn't. I didn't say it last time. I probably should have. This is one of those things where we tend to be water deprived. Okay, all land creatures, animals are um, uh, water deprived, so to speak, and we have mechanisms within our body to try to conserve that water so we can live outside the water. First life on the planet was in the water, and then when things began to start living up on land, they had to develop these mechanisms to continue to retain water in the body, and one of those is to have solid feces. Okay, some have solid feces, but most of your aquatic animals do not have solid feces. Most of your aquatic animals actually have liquid feces as well as a liquid urine type excretion. So, because they don't have to worry about conserving water. But land-based animals are going to have solid feces because we got to conserve that water. Okay, unless you're sick and you have diarrhea, you've got solid feces, and that's why diarrhea can be deadly. And what's dangerous to have it for an extended period of time because it will cause dehydration. because that water needs to be retained back in the body. So your intestines have these little villi, little hair-like, finger-like, hair-like projections that stick out, and they absorb that water back in. They also absorb, along with that, all the stuff that's dissolved in that water, such as your B vitamins that are made from bacteria that live in your intestines, and other minerals and nutrients that have been dissolved, digested out of your food, typically by those friendly bacteria that help us out. We have a lot of bacteria in intestines. That's a future topic, so I'll stop there on that. Okay. But that's going to be osmosis. Okay. And again, those little projections are increased surface area for more absorption so we can get more of that water out of there and keep it where we need it. So a solution is a solute and a solvent. Solute is the substance that's dissolved in the solvent, and solvent is the substance doing the dissolving. Here it says a fluid, such as water, that dissolves solutes. And generally speaking, that's the case. And for this class, you can say that a solute is a solid and a solvent is water. Okay, so you're dissolving some solid like salt or sugar in water, the solvent. That typically, I mean, for this class, that's what works. Okay, you can think of a solution that way for this class. However, I want to caution you that that is not the only type of solution you can have. If you go on and take a chemistry class, or if you've already had a chemistry class and you're learning that wait, solutions can be much more varied than that. You're right, they can be. You can dissolve a liquid in a liquid, fluid in a fluid, basically. You can actually, there are a couple of instances where you can dissolve a solid in a solid. That gets really weird, and that's chemistry. Okay, but for this class, water is the universal solvent. And those old alchemy guys, that's what they thought, was that water dissolves so many things. They thought it could dissolve everything, and eventually they found out that didn't quite work, but it still retained that name and that idea of the universal solvent because it does dissolve so many things. And for life, for living organisms, it is the solvent. Okay. So our types of solutions, there's three types of solutions you need to know. We have isotonic is the one we're going to start with. And iso means equal. You've seen that prefix before, probably in a geometry class. And you have your isosceles triangle. That's the equal triangle. Okay, equal. Iso means equal. Tonic has to do with the word tonicity. Okay, which has to do with water. So we're looking at equal water, and we're looking at equal water concentrations. So the solution, if you take a cell and you drop it in a solution of some sort, drop it in a beaker with a solution in there, the concentration of water inside the cell and the concentration of water in that solution outside is going to be equal, and that means that water flowing into the cell is going to equal the pace of water flowing out of the cell. Equal back and forth, isotonic, equal water. So the cell's not going to gain water, it's not going to lose water, so it's going to maintain that same amount because your flow is constant and equal. And that's ideal. Our blood cells are 0.9% saline, equal to isotonic to a 0.9% saline solution. So if you were to drop a red blood cell into a 0.9% saline solution, the cell will be happy. It won't change. There won't be anything drastic happen to that cell. It'll just keep on doing its exchange. It'd be great. And when I taught micro, I had my students do this with their blood. They'd prick their finger, put a drop of blood on microscope slide, and then I have them put a 0.9% saline solution on that slide, observe it through the microscope, and sketch it. Okay, and that can stay as long as the slide doesn't dry out. That blood cell will stay in that. They had all the time in the world. They wanted to spend sketching that, basically. And then we went through these other solutions, and then things got a little different. Okay. Um, the next I'm going to talk about is hypotonic solution. And this would be the last one I'd have them sketch because otherwise they have to prick their finger twice, but I'll explain that in a second. So hypotonic. Okay, here's where things get a little bit weird for me, I think. I was taught this to look at it from the perspective of water, because water's doing the moving, right? Water's moving and carrying stuff. So where's the water more pure and where's the water less pure? But the textbooks never teach it that way. Okay, they always use this other definition. So I'm going to read this first without the parenthesis there, and we'll talk about that. And then I'll talk about the parentheses, okay? So hypotonic solution, lower solute concentration than the cytosol of a cell, causes cell to gain water by osmosis. Okay, that's the definition of your book, or pretty close to the definition given by your book right there without the parenthesis. Uh, so lower solute. So that means that if you drop your cell into a beaker, the solution in the beaker has less stuff dissolved in it than what's dissolved in the cytoplasm or the cytosol, which is the water solution cytoplasm, okay, inside the cell. And that water is going to try to establish that equilibrium. It wants to have the same concentration of water outside the cell as inside the cell, or the same amount of things dissolved in it. Here, that's what we're talking about here, and dissolved inside the cell. So if you've got a lower solute outside of the cell, then things are going to flow to try to dilute the inside of the cell to equal what's outside the cell. That lower, kind of lower it. So the cell's going to swell. It's going to gain water, and if the discrepancy is too big between the two, it will burst. The cell will burst, pop. Okay. Let's talk about this from the water perspective. It's flip sided, same coin, heads and tails, depending on how you look at it. But so if the water is flowing from an area of high concentration to low concentration, and you've got less stuff dissolved in the water outside the cell, then the water's more pure there. So it's going to go into the cell to try to dilute what's in the cell to that same level of concentration. Okay, dilute it to bring it up to that same level of concentration. If you're looking at 0.9% saline and maybe, point, I don't know, 0.87% concentration, something like that, that might be able to distribute out, and the blood cell might be able to swell a little bit and not break, but it's got a very low tolerance on that. Generally speaking, anything you stick in there that's different, it's gonna break. Okay. All right, so let's look at the next one. Hypertonic. This is where you have more stuff dissolved, higher solute outside of the cell than inside of the cell. Same thing's going to happen. We need to try to get the concentrations the same. So the water is going to flow out of the cell to try to dilute what's outside the cell into the same equilibrium, right? Um, Generally speaking, there's not enough water in the cell to do that. Okay, so let's uh, let's look at this with the blood cell example. So I'd have my students prick their finger, drop a blood sample onto a microscope slide. I'd have them put the 0.9% saline on there, isotonic solution, everything's good. Then I would give them a like 0.2 or 3% salt solution, make it pretty high because I want it to happen pretty fast when I sit there and wait for it. A 2 or 3% salt solution, and they drop that on there. All of the water is going to run out of the cell because the cell's 0.9. It's going to try to put enough water out there to lower that 0.2 or 3% to 0.9, and it's not going to happen. Okay, so but what'll happen is it'll lose all of its water and it shrivels up into kind of a little. It has like crenellation is what's called crenellated cell. It has these little. It just shrivels. Okay, and so it kind of looks like my fist or little points sticking out on it because it's all just crumpled in on itself. And it can live that way for a little bit. It can tolerate that condition for just a little bit. So I tell the students they had to sketch all this. I said, make a quick sketch. You can come back and make it pretty later, but we don't want to leave the cell in that condition very long. And of course, there's lots of cells we're looking at, but they only had to sketch one. Anyway, then I had them put the isotonic solution, the 0.9% solution back on, because that would bring the cell back up to its normal state. And then when it got equilibrium established, then I'd have them put distilled water on it. Okay, distilled water is absolutely pure. Nothing is dissolved in it. It's all been steam distilled out. So now we've got 0.9% inside the cell, and outside you have totally pure distilled water. So water is going to rush into that cell trying to dilute that 0.9 to pure, and that can never happen because you got stuff that can't ever be pure. Okay, you can dilute it down to 0.000001%, but it's still never going to be pure. And the cell membrane can't expand that much. So that's going to cause it to pop. Okay. So anyway, students kind of always enjoyed that. It kind of, got this little shriveled up cell, and the next thing you know, they're blowing up the water balloons, pop, 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 all over the place. And so kind of fun. Uh, if you do in that order, you only have to prick your finger once. Otherwise, you got to go back and do it again for the next one. So hopefully you can kind of see there um, the two ways of thinking about it. You can look at where the water is moving based on purity of water, because water moves from high to low. And that's how I was taught to think about it. For me, that's the most logical way to think about it, but possibly because that's how I learned it. Yeah. And so if you were taught from the solute solution, or the solute, higher and lower solute, that works. That's great. Okay. For some reason, that's the way all the textbooks are teaching it now. And so um, stick with the way you know it if you've already learned it one of these two ways. If this is your first time hearing it, pick the one that makes more sense to you. Okay. And if they're both equal, probably go with the one the textbook's teaching, the lower solute and higher solute. Because I think that there is an actual reason for teaching it that way as far as going on into other sciences and looking at chemistry aspects and applications. Okay. Um, but and most of these, this in past when I've done a lot of short answer questions on tests, this has been short answer. You have to like describe, and so you had to describe it to me using one or the other. Now it's multiple choice, so it's not going to be maybe as much as having to write out your own answer on this. But don't try to learn it both ways. Okay, don't try to learn to describe it to me first in solute perspective and then turn around and try to describe it to me in a water perspective. Pick one and stick with it. Okay, because I've seen students do that and they usually get themselves twisted up on the test. So just pick one, whichever one works for you. Okay, but there are kind of heads and tails, flip side of the coin to way of looking at it. Okay, so isotonic, hypotonic, hypertonic, your three solution types. So we have passive transport, okay, which doesn't require energy. And as long as you're going from that high concentration to low concentration, you're going to have this passive transport. It's going to not require any energy. It's just going to go with the flow. I talked about how some things can get in the way of that. If you've got a charged particle, um, it could get caught in the tails of the phospholipids. So we have some other mechanisms. Okay, the channel protein, the carrier protein, those things can help get those into the cell. Since the channel protein is often analogy is a bridge, students usually don't have trouble with that one, thinking of that one as passive. Okay, but the carrier protein sometimes seems to be problematic with students in that they want to think that requires energy. Because I use it a ferry boat analogy. And if we think of a boat's got a motor, you have to paddle or something, right? Um, but as long as it's going with the flow, it's more like instead of going across the stream perpendicular to the current, we're actually going with the flow downstream. Okay. If you're going with the flow, high to low, downstream, it doesn't really require a lot of energy to go downstream. Okay. So it's still a passive. Now it's got a specific term applied to it, which I'm going to get to next slide. Um, but active transport is obviously the other type of transport, and that requires energy. So these carrier proteins, the type of transport that they provide, it's still a passive transport, but it's referred to as facilitated transport. And again, facilitated seems to imply sometimes that there's something working, and therefore the idea that it may require energy gets into students' minds. But look at that last statement, does not require energy. Okay, this does not require energy. So an analogy I've always used, okay, is to think about being on a canoe trip. Okay, we're on a canoe, not other type of boat, not a raft. Okay, because that I last two classes today, the raft has come in as a as part of the discussion. We're not talking about rafts or other types of boats. We're just looking at a canoe. So think about a canoe and how a canoe is shaped. And you're on a float trip down the river, floating with the going with the flow, right? And you and your buddy are in the canoe, and buddy says, "Hey, see that bend down there in the river? Let's have a race. How about you get out and float on your back? You're not swimming, you're floating. Okay, float on your back, and I'll stay in the canoe. You don't swim, I'm not going to paddle, and let's see who gets to that bend first. Who would you project would win that race, and why? So just give me you can say it's just gut instinct if you don't have a reason why, but if you got a reason why, tell me why you think that might be. So what do I have? opinions, ideas? Everybody went to sleep.
I would say the canoe. That's my opinion.
You say the canoe. Okay, why?
I really don't know. That's just what my instinct says.
Gut instinct. Okay, that's good. Works. Yeah. All right. Anybody else have same idea with maybe reason or different idea? Okay. Well, you're right. Okay. Yeah. Um, And the reason, theoretically, at least you're right. Okay, we had got into a discussion of, well, but I've had friend fall out and they got swept down the river faster than I did, and that sort of thing. And I'm like, well, I'm not talking about raging rapid. And then it did come out, oh well, okay, they were talking about a raft, not a canoe. And so yeah. Anyway, uh, the canoe is designed to kind of move through that water with less resistance. Look at how the front of the canoe is shaped. Okay. We are not shaped to move through water. Okay. So we would provide a lot of resistance. And so that's what the carrier protein is like. It's designed to be able to move through those phospholipids without resistance or with very little resistance. Whereas the molecule trying to get through there on its own could get tangled, it's going to have resistance. So uh, we're thinking about carrier protein as a boat. That's kind of what you think about it as it's a canoe. Okay, it's going to go through with less resistance than would otherwise be encountered. So that's how it facilitates the process without any energy. Okay, because all your friend is doing is maybe steering to keep the canoe from bumping it, veering off into the bank or something. But no paddling, nothing going on, just going straight down with the flow. Okay. Now, if you turn around and you paddle that canoe upstream, that's a totally different story. That's going to be active transport. It's going to require a lot of energy. And that's not achieved with the carrier protein. Okay. That's going to be achieved by something called a pump. And we won't get into that anymore in this class. This is your only time you're probably going to hear about that in this class, is to just know that it exists. Pumps. Um, my botany students, I told my students last semester, I said, if you take botany, you'll wind up talking about pumps. And I've kind of already seen where that's going to show up in the book. It's with leaves. We study leaf tissues because there's some pumps, tissues that have these pumps to control the opening and closing of the stoma, which are what regulate gas exchange. Human anatomy and physiology is not my forte, so I don't usually give an example there because I don't keep one on the top of my head. But if you were to take human anatomy and physiology, then in the physiology aspect of the class, you would be talking about some of these pumps that exist in certain tissues in our body as well. Okay. Um, but they require energy just like any pump. If you think about a pump that's on a well, it requires electricity or something to run it, or if it's a hand crank, if you've ever done one of those, you can pump one on one of those. Seems like forever before you get water out of them. And so a lot of energy required there. A lot of energy goes into that. And these are usually pretty heavy energy consumers in the system when they have to operate. But that energy is required by supplied by ATP. Okay, we talked about that back in our chemistry chapter. Adenosine triphosphates are kind of our cellular battery. And so that's what's going to supply the energy for this. And a lot of this transport is large molecules, okay, that can't be transported in as well. And that's regardless of flow, regardless of gradient. You get these large molecules that just can't fit across the membrane. They can't even be carried on by a protein. They're just too big. So they're going to have a vesicle formed around them. And that you saw that process, it's endocytosis. You saw that in the film, the virus film. And I think it was one of the questions I had you write endocytosis down, and it wasn't said very clearly in the film, so you kind of almost had to look that up. And I think I hinted at it too beforehand. But when that virus particle attaches to that protein that it attaches to, and then the cell just kind of in the video, it just shows it sinking into the cell and this little pocket formed around it. Okay, that's endocytosis, and that requires a lot of energy. That is an energy user to get all that going on. Okay, looked pretty simple, but it's not. And we have two types of endocytosis. That process is referred to as phagocytosis because that's a large molecule being taken in. And where we actually first observed that was with macrophages. And these are some of our immune system cells, and they engulf bacteria that's in our bloodstream. And also, they go around and they're also a little bit of house cleaners. They will engulf worn out red blood cells. So, talked about those red blood cells popping. Uh, they also just wear out. They get old, they wear out, they break down, and they get replaced. But the old ones have to be cleaned out somewhere, or they mess their, gunk everything up, right? So macrophages, that's part of their job as well. So that's endocytosis of large molecules. It's a specialized name we give that. We have another specialized name that we give to the taking in that same vesicle type formation of liquids or small molecules such as proteins, and that's penocytosis. The reverse or opposite process is exocytosis. This starts at the Golgi apparatus, which we haven't really talked about our cell organelles yet, and we won't until we get to the eukaryotic cells. But hopefully, you've had these in life science and you've had them in biology in high school. Um, the Golgi apparatus, it's also called Golgi body, which is how I first learned it in high school. And in college, it had become the Golgi complex, and now it's the Golgi apparatus. It's like they can't decide what to call it. In a fill in the blank type question on a test, I would accept any of those three. Okay. Um, but Golgi apparatus is the current term. So that's the one you should learn if this is new to you and you haven't learned one of the older terms. And then if you can replace one of the older terms, then in your mind, do it. Okay, Golgi apparatus. Um, this is an area that makes these little vesicles inside the cell. And in your drawings, in your book, in your cell diagram, they're usually depicted as little, my hands up here, not at that angle, um, kind of stacked pancakes. Look like little stacked pancakes. They start off larger and get smaller, and they're curved. Okay, look like a stack of curved pancakes. If you see that, that's how we always depict the Golgi apparatus, and they're shown in different sizes because they're being made, they're being formed, but they're also just waiting. Okay, the larger ones are out there just waiting to be needed. That's often referred to as the packaging and shipping department of the cell because whatever needs to be shipped out of the cell is going to be stuck into one of those larger completed vesicles and then shipped out. And when that vesicle's got something in it, it's going to move up toward the cell membrane. And then those phospholipids, they merge with each other. So it's just going to merge right in. I'm going to move it out to here so I can do the next step. And then it just opens up and folds out, and whatever's in it is released out of the cell. Okay. And that cell, that becomes part of the outer membrane of the cell then. And so sometimes students will hear that and they go, oh, that's how cells grow then. They just, all these things getting left out of the cell, released out of the cell cause the cell to increase in size. But that's not what's going on because while we've got things being released out of the cell on here, over here, we've got something being taken into the cell with endocytosis. So it's this constant ebb and flow. So the cell membrane maintains the same average size. But that again is part of that fluid mosaic model we talked about. That's part of that fluidity of the cell, okay, of the cell membrane. All right, so um, those two processes, merging and exchanging and going on there. So a specific term that's applied to this is if the product of the cell that is in that vesicle that's being released is a hormone, then this process, this type of exocytosis is called secretion. Okay. Um, anything that needs to get out of the cell can be sent out that way. Any large particle. Waste products are also that'd be excretion, term for that. But exocytosis is how those can be removed as well. Okay. Okay, a little bit of change on topic then, still in the same presentation though, but I'm switching over to uh, bacteria here. We're going to start talking about prokaryotes. So to refer back to chapter 1, where we had our taxonomy classification stuff, we've got two different types of cells. We've got prokaryotic cells, we've got eukaryotic cells. Um, Eukaryotic cells have membranes and membrane-bound organelles, and we're going to talk about those later. We're starting now with our prokaryotic cells. These are cells that don't have membrane inside, other than to have maybe just some layers inside that outer membrane. Okay, no membrane bound organelles that have very few organelles at all, actually. Okay, so um, those are the earliest cells that we have fossil evidence for, and if you look at things logically, that would be the first type of cell that would have come into existence anyway, because it's the simplest. Okay, so it is just membrane, genetic material inside, and some proteins that are needed to replicate it. Then, of course, you've got the membranes that are needed for burning the sugar. Okay, and but those are layered. I've got pictures of that. Won't get to them today, but I've got some pictures of the membrane folds that I'll talk a little bit about today. But I'll show you a picture probably on Friday, so we'll get to that, I think. So I've already mentioned about the cyanobacteria. We talking a little bit more about that here in a little bit. Um, about how it was the uh, first known cell on this planet is the cyanobacteria. It's a photosynthesizing bacteria, and that is what put oxygen into the atmosphere of the planet. Okay. Um, so those are kind of review slides of things I've already said. Here's a second picture of a stromatolite. I had shown you a stromatolite on Monday as well. This is another one, and I may have kind of skipped over and shown this to you on Monday. I can't remember if I did or not, but this is a picture of a cyanobacteria today in the ocean, still growing in these mats, and um, you know, potentially we've got fossils building right here in a sense that um, uh, sediment is going to fall down on these. These are going to die, and new cells are going to grow on top of the old cells, and the sediment mix, and you get more sediment, and enough time and pressure. These lower levels down here will maybe someday become fossils of the future. Okay, so this process has been going on for billions of years. If we gram stain, which again later I'll talk to you about the process of gram staining and the different shapes of bacteria when we get into that. Um, I do that with standard bacteria. I don't talk about that much with archaebacteria. Uh, gram negative, it means they're going to under a microscope they're going to look pink, kind of red pink. We call it red. Students usually argue that it looks pink, but it's red. Okay. And rods, which the proper term for that is bacillus, and it's a jelly bean shape, kind of a long jelly bean shape. Uh, that's the descriptor of what they look like. And they can photosynthesize. Again, we used to think they were blue-green algae. Uh, they do have chlorophyll. All of them contain chlorophyll, a green pigment. You cannot photosynthesize if you don't have chlorophyll embedded in your membrane. Okay. Um, they can also contain other pigments. All right, like red, yellow, brown, or black colors of pigments. And what it is that you still have the chlorophyll, but if you've got some of these others in larger quantity, that's what you're going to see. You're going to see the one that's the pigment that's the most numerous in that particular cell type. Okay, so we still consider them all blue-green, whether they're that color or not, because they have the chlorophyll. And the ones that we identified first were ones that had mostly the chlorophyll, so that's what we saw. But they're still related. These are all related to each other in their function and genetics and things like that, just pigment differences. So these are pretty large cells. They're 1 to 50 micrometers in width. What's a micrometer? A micrometer is very small. How small? If you take out a metric ruler or take out a ruler, because your English has metric on the other side, just make sure you're looking at the side that has the centimeters on it, centimeter marking, and like look at the distance between numbers 1 and 2. It's about between my fingernails here. You can see that distance. Okay, that'd be about what it is. And if you look closely at that, then that is divided into 10 smaller marks. Well, each of those 10 smaller marks represents a millimeter. So then if you go down and you look at the distance between those 2 millimeter marks, just pick two of them and look at the distance between. Now imagine dividing that 1,000 times. Okay, there you go. All right, your I'm sorry, your micrometer. It's very small, but we can see those things if we magnify something 1,000 times in a microscope, which your high power oil immersion lens is 1,000 magnification. We can see that. Okay, and we're not looking at a micrometer here; we're looking at up to 50. Okay, so these are the same size as some single celled organisms. Some of there are algae. That we could see single cells under the microscope. You can actually see them under 400 power, though they're really really small. Um, and so that's why, like I said originally, we thought they were algae. With even higher powered microscopes, though, we got to see inside and find that they do not have organelles. So that's why they got reclassified. Prokaryotic cells, or they don't have, I guess, specify membrane bound organelles. There's one organelle that doesn't have a membrane on it. It's called a ribosome. I mentioned that already, previous lecture sometime back. And all cells have that. Prokaryotes, eukaryotes, okay, they all have that particular organelle, but it is not membrane bound. And that's the difference. Prokaryotes can have organelles, they're just not membrane bound. Eukaryotes have membrane bound organelles. Okay. Um, hey, let's see. Did I? Yeah, okay. Um, so archaebacteria. I feel like I'm ahead of myself, but I think time wise we're good. I feel like I skipped something with y'all. I'm not sure why. Okay. So Archaebacteria often live in extreme environments, and they're sometimes referred to as extremophiles. They don't have to be in extreme environments. Some of them live in normal, moderate environments and fine. Cyanobacteria are pretty versatile, and they've got some ranges in what they tolerate. Different types can tolerate different areas, uh, and you can find cyanobacteria in some hot places, but you can also find it right down here in Grand Lake, okay, which is a normal environment, not an extreme environment. Um, I can go to this picture next year. Uh, this is the Grand Prismatic Spring at Yellowstone. Um, I have seen another picture. I have some other pictures of it that show it in much more vibrant color. I think it's just kind of angle and all of that. This center out here is usually a pretty dark blue, and it is boiling. Okay, nothing lives there. But as the water cools off toward the edges, you can start to see this cloudy green appearance. That's a cyanobacteria growing there. Okay. Um, this is another cyanobacteria out here. This is a yellow one. Okay. And then we get into this kind of brownish one, and there's a further on out, I think you get a little bit more orangey look. And like I said, I've seen some pictures where we get a little bit better representation of the spectrum of color changes in these cyanobacteria that are growing here. Cyanobacteria are specific, some of them are very specific in the temperature range they can tolerate. And if you know what temperature range they can tolerate and which pigment they are, you can actually tell what temperature the water range is here based on the colors of cyanobacteria that are growing. Okay, which is kind of cool. Uh, so out here on this very edge where you've got this kind of brownish, there's a uh, this picture is somebody I think took this on the walking path. Okay, there's a raised wooden walking path that you can walk on here. Uh, and I remember putting my hand down close to the water. Okay, and I could feel the heat coming off it. You could see the steam, and this was summer. You could see the steam coming off the water a little bit, but I put my hand down close to it, and you could feel the heat. But it wasn't horribly hot at that point, and there were little water creatures uh, living they were swimming around in there, okay, like little like insect type things. So there's some things that it was warm, but it wasn't too warm for that. So it cools off as you get out here further away. um, so like I said, temperature ranges are um, quite wide with some of these. Okay, but at Yellowstone you also find that you've got these hot springs that are very acidic as well as hot. You get the sulfur. You can smell them, when you approach them. Um, and so here we have acidophiles. I don't have that term on here, but acidophiles are acid lovers. So you have some that like acid. The halophiles, they like extremely salty places. Thermophiles, that's what is in that hot spring there. They like the hot water. We have some that like frozen too. And there's another term for that, and I don't ask you that one. I don't think on the test either. I've got it. I'm gonna have to go find my document because it's not up. I thought I'd put it up on your um, module. but I have a document that goes into more of these definitions, these descriptions, okay, descriptor terms for these extremophiles. But we find some of these living in the Arctic and the Antarctic ice near the poles. Extremely cold. They love it. Okay. Uh, so methanogens, have a story to tell about methanogens. So I'll have to start there on Friday. Okay, because I'm pretty sure I'm out of time. Close enough. All right. Questions? Okay. I will see you later then.